geographic models of virus migration. This is work that we've conducted at Arizona State University. My co-investigator is Matthew Scotch, and it was main author by Davey Wischenberger. He's not here, but I'm presenting in his place. So phylogeography, it's science that studies virus migration and mutation. And basically, it looks for specific patterns where viruses are transmitted, particularly zoonotic viruses, viruses that are transmitted between animals and humans. It is very important to be able to track the geography of these migrations and mutations, because you can make interventions on time. You can trace back to see, for example, if there is a specific group of animals that are moving around or are traveling. For example, it's very common, for example, to see, at least in the US, these petting zoos. And sometimes there has been models that have traced back patterns of infection to how these petting zoos are traveling through many rural locations. So it is important to have accurate information about the geographic location of the different samples that are collected. So that takes me to explain a little bit about the significance of our work. So the tools of phylogeography are basically databases. So there are people, of course, that do collect the samples for their study, but this is a collaborative environment, and many times the people studying this migration and mutation are not the same people that are collecting the samples. So they rely on databases such as GenBank to track this viruses, the virus spread. They can go across continents. They can go across many different countries. So it is vital for them that the information that's found there can be used. So to do any of these studies, they will take the genetic sequence and the collection data. So the host, the time, the place. The problem is only about, per our calculations, only about 20% of geographic location names, toponyms, in GenBank are precise enough for phylogeography. Could be traced back to nomenclature. If you look at GenBank, there's a field that is called country, and that's where people are supposed to put the location of the sample. So, of course, you think country and you put China, not thinking that that's kind of big to do any modeling, or United States. And some people actually do that. I mean, it's just ask of a country and they put the country. Others, of course, put more detail. And we found that it's actually not always in that specific field, but the sequence name has embedded in it some specific geographic location. So this study that we're presenting now here is to resolve toponyms mentioned in publications using other knowledge, okay? There's 2 points of view to this problem. One, of course, starts with GenBank. If you have a specific record that you're interested in, or a set of records that you're interested in, you want to get the specific location for those records. And for that, you go and enrich with the literature. The point of view of this paper, however, is the other one. When you have a paper, there's geographic locations in there, and you need to disambiguate them, okay? It's separate work. You might think that it's the same, but really it's not. And the point of view that we're presenting here has wider applications beyond phylogeography, because of course there's many papers that do not refer many times to zoonotic viruses or even phylogeographic studies, and you still need to disambiguate these geographic locations. So you get a mention in a paper about an H1N1 virus isolated in 2009 from a child hospitalized in China. So you have that segment of a sentence or a complete sentence in a paper, and you want to enrich and use other knowledge, particularly metadata in GenBank. We built the model so you could use other things, not only GenBank. So that's why I keep referring to knowledge enriched or using other knowledge. So it's not just metadata, but in this particular instance, that's what it is. So we found a matching record, which again is not always straightforward. We use other information like the organism, in this case H1N1, the host, the country, China, which was contained, the mention in the paper had China in it, but I'm trying to use the pointer here. Let's see if I can. Had China in it, but not any more specific information. So by double checking, many publications have more than one GenBank record that it refers to. So it's not a one-to-one match, there's many. So you have to find the most adequate one. In this case, our engineer points out Nanjing as the more specific location for this collection site. So toponym resolution then will in the end assign a unique coordinate to mentions of geographic locations. So it's not enough to have the place, you need the specific coordinate in order to be able to study these things or to use all this information in another study. So there's two steps to toponym resolution. We separate them, detection and disambiguation. Detection is basically finding it in text, okay? So simply matching that this Nanjing and this may be both mentions of locations, or if they go together or not, they not always go together. Then Sanger here over here. The idea is for disambiguation is which Nanjing are they referring to? There's 4 possibilities even within China. And for Sanger, we have an area in California that does that name, and a scientist name, of course. So we have to disambiguate, try to find out exactly what this specific mention is referring to. We use known detection methods in this work. We did not focus on detection so much. As many of our text mining efforts, we started with the idea that this was solved. And not so. I've learned project after project that there's nothing really solved in text mining. So, well, but we still have a place to start. So we started with the dictionary-based approach, basically just find the exact match with a given suitable lexicon. In this case, there is a GeoNames database. It's the most widely used, it's the biggest. It has over 10 million names and it has really, really broad coverage. It has, of course, advantages and disadvantages precisely because of the broad coverage. It creates a lot of ambiguity, so any match is a multiple match. But at least for detection, you have a name and then you might think that it's a location and then go to the next step with that. Then others have proposed some rules-based approach, of course, where you learn the syntax and patterns of how people mention location names and get a stop word that you eliminate some of. So although is a place, it's in GeoNames, so we need to not have it there, otherwise it's going to create all kinds of extra matches in a document. So for disambiguation then, there's, again, other approaches that have been proposed before us. Use of lexical context, and actually many of this, of course, if you're familiar with text mining, are similar approaches to any kind of disambiguation. So lexical context helps, where you look at other locations or places named or mentioned around the one that you're interested in. Then we move into into the what I'm referring to as knowledge area, where you're not using any of the text present in your text mining problem, but you're using some additional resources to help enlighten your process, in this case, disambiguation. So here, others have proposed a population heuristic, basically given choices, choose the most populated place. The logic of that is basically a lot like statistics, okay? So it's likely that it's going to be there, the biggest place rather than the smallest place. You might disagree with that one, but it happens to work. So, so that logic doesn't always work, but it actually works most of the time. Then the distance heuristic, we choose candidates within the smallest possible geographic area. And for this one, we start looking at why does it matter that we look at the whole paper versus just looking at one GenBank record and trying to disambiguate just with that. With this one, we look at the larger context, that all locations mentioned in the same document, and try to plot them and sort of connect them and find the smallest area possible if you connect all the dots, all the places mentioned in them. It also helps disambiguate, because it is likely that in a study, you are not getting things from all over the world, but just one area. Again, one of these things that logically, it might not work for all cases, but it works for most cases. So if we keep adding to it, so what we're adding, and we tried adding here, is just metadata, and then use some other knowledge either present on the paper or present via combination of other sources. So we have tables in the document, we have metadata in GenBank, so various fields in linked records of GenBank. We also try and are experimenting with using other papers published by the same author, other GenBank records referring to the same paper, and some other collection of knowledge that can help us disambiguate. A quick overview on the pipeline then that I've been mentioning. So given a PubMed article, okay, we detect all the mentions of specific places by matching geonames, and actually we use an additional database when we discovered, when we did some error analysis, and we discovered that geonames was really good at the specific names, but not the common names of places. So Republic of Korea is in geonames, but Korea is not. So on its own. So we had to adjust for that. And you don't want to always take a subset of a match and call it a match in all cases. So it gets tricky, as always. So we have that, geonames, another database, and rules, get that detection going. And then once we have that, we get the coordinates from possible coordinates in geonames. In this case, it will give several matches for Quebec. And then in disambiguation, we start looking around in the text and apply all the other different levels of knowledge that we learn from other people that have tried this, plus the ones that we added, and try to get the best match that we can. In this case, we found that the strain, the country field in the GenBank record had Canada in it, but the strain of this in the same record had within it the name that we wanted, Quebec. Okay, so we eliminated the other coordinates and finally settled on that one for our match. Parts of our contribution for this work, we created the gold standard, is 60 full-text PubMed articles, over 500,000 tokens that were annotated. We had a team of 3 annotators, and we did different subsets of annotations for this papers. For these 60 articles, we got 5,730 GenBank records that were linked to them. 99.9% of the GenBank records had some geospatial information, but 74.5% of this 5,000 were not precise enough for a phylogeography study. This sample, this was H1N1, and we had flu virus as well in other parts of the study. Is that 3 minutes for the 20 or 3 minutes?
3 minutes for the 3 minutes.
Okay. So for annotation, I wasn't sure what you were signaling. For annotation, these are just details that you can find. It gives you an idea about the task. On average, we had 19 ambiguous candidates per toponym. We had extremes, like San Jose has 676 matches in geonames. We reached in detection an inter-annotator agreement of 97 in precision and 98 recall. So we had pretty good, we were pretty confident on our annotation process. Results For disambiguation, detection only, as I said, we use prior work, so it's not anything that we're highlighting right now. For disambiguation, the population heuristic, what was previously published, reaches 0.75 precision, whereas the distance is 0.62. When we mix these two together, plus the metadata that we added, we reach 0.89%, okay? Of course, all of them, since it's disambiguation only, we're given all the mentions. So recall is just an artifice there, so I'm not emphasizing F measure in this case, just precision. For both tasks combined, of course, having low precision, low performance in the detection task affects the detection and disambiguation overall task. But we still see some improvements, okay? So 0.55 with adding all the knowledge together. We have the gold standard, it's available, you can download it. We also created this evaluation of a new model for toponym resolution, utilizing alternative knowledge sources, particularly metadata, but it's prepared for other layers as well. And that overall it outperformed common heuristics such as populations and distance. We have ongoing work to address the limited performance due to toponym detection. So yes, obviously we went back and are working on that as well to improve the overall task. This is an example of what we're doing for that task, just basically throwing in some machine learning. And then completing the full cycle. So we are now working on the other end of the spectrum, enriching specific locations in GenBat, using all available knowledge sources, including all other locations mentioned in the paper and other papers that refer to that sequence and so on. And then another one is like, okay, so we're improving all this. What's the end result? Some quantitative validation of the contribution of our contribution of more precise locations to the models themselves, okay? So if we improve the locations, can we improve the end result, the models of migration? That requires much more additional work by doing statistical analysis of the different confidence levels in the locations and throwing them into the models and seeing which model results in the best performance or more accurate depiction of the actual model of migration and mutation of the viruses. Okay, so that concludes. Funding for this work is NIH, the National Institute of Infectious Diseases, and my email, if you need to contact me. Thank you very much. And I think I have time for questions.
Questions? Questions?
Have you ever tried to do the detection by using the machine learning system? Because now you have the gold standard data. This is like a car problem in text mining that just name entity recognition, and the machine learning will outperform all the dictionary base.
Yes, I found that many times it is the case. We have tried machine learning. We are in the process of finishing some experiments on that. We had the problem with a lot of ambiguity in this one. So the machine learning basically got trigger happy and started tagging everything as a location. So you need to adjust a lot because of that. So it's a highly ambiguous space. But yeah, we are experimenting with that.
Yeah, I have worked on kind of similar case, but it's just detecting cell lines. So if you use machine learning and then you, let's say like use machine learning and support the machine with the dictionary base, for example.
Yeah, no, yeah, I've seen that before in other tasks, in genes and so on. So I've used that before and convinced that we can make it work. But we're working on it.
Thank you.
Thank you.
Other questions? Comments? I have one. Have you looked at all at the resource? So you've been trying to do curation from the PubMed to try to identify where they are located. So now there are two United Nations organizations that are actually looking at circulating virus. That's the Food and Agriculture Organization in Rome and the World Health Organization. Have you actually contacted them to
No,
show
we
them
haven't.
So you should
But it will be good. It will be
it would
good to do that.
Because there are, so most of the time when a virus is sampled into the wild, the first thing, it goes both to WHO if it's a human that got infected, and that is tracked. Then the sequence goes to the sequencing center, and then we need to bring these two together. The reason why I'm talking about this is because the MPRSI, which is the early risk epidemic from the FAO, has been linking both viruses on the sequence side as well as the isolates that were sampled. And they have the geolocalization because they send either people there or they have actually the GPS coordinate. And this is a result that have about 7,000 or 8,000 viruses that have been linked.
We have found that in this field, there's many personal communications. So before the sequences and the places make it to the databases, there's many papers actually published saying we contacted the people that collected this and they told us it was this. Even if, so we're trying to accommodate for things like that and see if we can incorporate some of this knowledge that is either in not databases that are not GenBank, or even personal communication by contacting the author. So it's just a matter of having the right interface. Yeah.
Are there any other questions? If not, thanks again.
Thank you so much.